So hello again to the very fresh audience, right? Just checking. All right, so I have 40 minutes to complete this session, and I don't think I can do it like I did in all the years. So I won't be able to wait for all of us to go through all the steps. So what I mean is that you guys are already pretty expert, and you've seen how to process the proteomics data. So the aim of this tutorial talk session would now be to not redo what you already know, but let's touch on the points that are critical for you to know by end of this talk. So what are PTM specific data processing steps, even if you don't go through every bit and piece of the exercise. So let's try, that's why it's going to run a little different than what I've done in the past years when I had 1 hour, 15 minutes versus this. It's just not possible. All right, quickly, the data set is, as you remember, made of, get the pointer. Yeah. So the data is made up of 3 experimental conditions. You got 6 controls, 4 samples representing mitosis, EGF 15-minute samples, 4 replicates there. Now, so far you know what all columns do you upload when you're analyzing proteome expression data set and the interactome data set. So I'll only talk about the columns that are specific to PTM analysis. So let's first talk about what do you upload in the expression columns. This is already where we start to be a little different. There is something called your condition followed by _1, _2, _3, and we then historically converted it to _1 because a lot of us like to call our conditions as _1 replicates. So you're going to have to fix that as a problem in general. So please do not name your files as __. Otherwise, we'll have to make a triple underscore there. All right, so what does it mean to begin with when you speak of 3 intensity columns reported for 1 sample? So this is actually representing data for the same sample, but the site information is derived from singly, doubly, or more than doubly modified peptides. So imagine, so if you remember now the tutorial, the lesson that we had before about the PTMs, Let's go back to the modified peptides. So you remember that a modified peptide can be singly modified, doubly, or more modified. And those quantification events have to be brought together into a site-centric view. So how would you handle this? And one way is choose one of the forms. But very quickly, as a biologist, you would know why you would not settle for a singly modified form versus doubly. Think about activation loop of MAP kinases, which has these 2 important residues, both of which, when phosphorylated, only then you know the kinase is active. So in that case, you're interested in following the dynamics of doubly phosphorylated form. However, you sometimes are interested in figuring out the cooperativity of some of these phosphorylation events. So you're also interested in knowing what happened at the site resolved view, even if it's multiphosphorylated species. So there is value in keeping information resolved to all the 3 levels, and that's why the information is left for the user to use whichever way and draw conclusions accordingly. Is this clear? Because this is one of the key differences in the expression analysis versus PTM analysis. And this is, again, site-centric view, and therefore you have _1, 2, 3. Now, the other thing that you load specifically for PTM analysis is localization probability. We spoke about how do we calculate this. And this is uploaded, obviously, as a numerical column, and you can filter for your class, your phosphosite data for certain localization probability that you think makes sense for you. We usually filter it for 0.75 or higher when we are reporting the depth of the proteome, depth of the phosphoproteome. However, that's not necessarily the case when you're performing the statistics. And why am I saying that? Because sometimes you may have events where you've not been very good in doing site localization. However, the trends for, let's say, less accurate localization may still give you some idea on if there is a modified peptide which could not be localized well, and there is a trend here that you can go and cross-check in your modified peptide table. So keeping some of those events here may be useful, especially when at the end of your whole exercise you learned 15 members of a apoptotic pathway have changed, and 2 of them you would have removed if you were not using the localization probability filter so high. The other important column is sequence window. So this is an output already sitting in your phospho-STY tables. What essentially sequence window does is puts your modified peptide at the center and reads 6 amino acids on the either side. So it's a 13 amino acid sequence window centering on your modified peptide. And you will see the advantages of this as you start importing data from the other data sets, databases. The other one is amino acids. So in case of phosphorylation, it will be serine, threonine, and tyrosine. So you want to know what is the amino acid that is getting modified here. And lastly, the position. So this is referring to the position of the site on the protein. And all these are features that are going to be extremely important once you start to import data from other databases. So these are exclusively seen in PTM analysis that you would be uploading at the start of your exercise. Is this clear? Are there any questions on this one? There's a question?
Yeah, numerical. The last one that you just mentioned, databases might report the protein with or without processing. So databases might report a protein with or without processing. So would it be numerically starting from amino acid 1, which is the initial methionine, and then continuing, or?
So it's all connected with the UniProt identifier.
Okay.
Whatever it is, this is for that UniProt identifier position on that protein.
Okay, so if you have like a nuclear localization signal tag that's removed, it doesn't start after that. Okay.
Yeah, the answer is back to the UniProt identifier. Yeah. And if we have information on, for example, isoform result and some of those things, so it's again going to be encoded in the identifier, and that's what MuxQuant uses. All right, so next one is preparing and processing your data. You would realize a lot of things, again, are pretty common. And this is my favorite thing to remind ourselves why we do what we do, and then we figure out, I think all of us will somehow figure out how to do what we want to do. So let's just go to different steps. There is a theme you've seen so far. As soon as you start with your data set, you've been doing filtering of reverse and contaminants. After which, in case of PTM analysis, you would filter for high confidence sites. So you will use your localization probability filter and keep those sites as you report the depths of your study. The next one is just like any other study. You would be doing log transformation of your intensity data. You would rename the column using regex as needed. The other very important feature for PTM analysis is expand site table. So remember I just mentioned about _1, _2, _3, which means for each condition you have 3 columns. This is very hard to start working or doing any statistics on. So one way to work on this data set is to convert this information of one phosphosite, 3 columns into so-called expand site table, where we collapse the quantification information for PTM sites into 1 column. So as you would go through the data set, you would realize that there is a 3 times increase in the number of rows because you've converted this information into unique identifiers, and your data becomes 3 times the number of rows. And concomitantly, you see a reduction in the number of main columns because you've collapsed the information in this dimension. So those are quick ways of checking what's going on and how you're following your data set. The next one is, again, pretty similar to what you do in your routine expression analysis or interactome analysis, you define the experimental groups, and then comes valid values filter. The rules are same, impute, but with good filters before you impute. Rule of thumb, usually half or more is good, or define the groups and apply filters based on the group level. The next step obviously is to do ANOVA testing, just like you do in the expression proteome analysis, after which you can do filtering for significantly regulated, do the regular downstream z-score clustering. Sometimes when we do these things as steps, it's hard to keep the big picture. That's why I just put it here, how you're preparing and processing your data. The steps are same, however, there are small nuances when you speak of PTM data analysis. Now, in terms of annotations, obviously you add all the annotations that are coming from different databases like gene ontology or KEGG pathways, and there are many others that are out there. However, for PTM analysis, now you can add many more features which are very PTM-centric. For example, are there site-specific features? In case of phosphorylation, you can start to ask, is there a prior knowledge in terms of which kinases do phosphorylate this? Is it already known to be important for a biological function? So all of those are very, very PTM site-resolved features, and there are ways to pull those into this data set. Those annotations are not included in any other data analysis workflows that you've gone through this so far. And lastly, there are other things which are not just site, but the site surrounding or site local environment, if I were to say. So you can talk about, is there linear motifs that talk about what would be the best environment for that PTM to happen. All of those are very, very interesting features, especially once you start looking for specific biological condition of interest. So let's look at some of this analysis, and I want to leave some time for us to discuss any specific questions in the analysis that we may have. In terms of enrichment, protein and site features, there are certain things that you have to keep in mind. Unlike your expression matrix, you have to remember that there may be different proteins represented in your matrix multiple times, depending on number of times you've seen modifications on them. So it's not equal representation of different proteins in your base matrix. So if your 10,000 proteins each have 1 to 10 phosphor sites, each of them is present different counts in your big matrix, which means you're not giving equal weightage to each protein. If a protein is overly phosphorylated just because you've counted it, let's say 100 times of your matrix, compared to something else which is hard to detect and there is only one event. So you're just counting that 100 times versus one time, and you have to keep that in mind when you're doing the enrichment analysis. So there are tricks around that when you're doing the enrichment analysis. So we'll have to reach to that step somehow. And lastly, sequence logos. So using the linear motifs and also maybe figuring out the sequence window corresponding to the sites that are modified, you can bring them together and generate so-called a sequence logo, preferred sequence context that is regulated in a particular biological context of interest, and that can help you, along with kinase-substrate relationship for that cluster, to start building the hypothesis around what are the kinases at play and what are the sequence features that are getting modified. So this is the kind of information that is exclusively to this data analysis, and they're all feasible in Perseus platform. So before we get into the little bit of these details, are there any questions about these? Oh no. Alrighty. So what I've done here is try not giving you a chance to do the exercise, just follow the session that I've already done. which would help us to also do any specific steps that you may want to learn more about. Come on. Please do ask a question while we fix this. Okay. Don't make it big. Okay. Yeah, don't touch it is the answer. Okay, so also will tell us how to walk through a session that is already completed. If you look at your exercise data set, we are uploading the intensities corresponding to different conditions. Please do not try to do this exercise. Let's just walk through this because it's just not possible. So let's just try to walk through this session and go back to the main questions. So you, as I mentioned, you will have, let's say for control 1, you have 3 different _1_2_3 that is uploaded. And then you have all the other columns like position or sequence window all already kind of default in Perseus to upload them in the right columns, but you should know why you're uploading them there. After which you do your you have your matrix uploaded with these columns. Always a good idea to keep an eye on the number of rows that are here. Will you give you an idea of when I filter, what is the impact on the total number of rows? After which you do your regular filtering for localization probability, as I spoke. You do it for any localization probability more than 0.75 should be retained, anything lower excluded, which reduces this data set of 60,000 phosphosites to 42,000. So that's an impact when you talk about reporting the depth of your studies. Again, I would not have done this, continued it here for the downstream steps, but for all the statistical analysis, I would still keep all your non-class 1 sites for the reasons that I just explained. Then you can do your filter for reverse and contaminant. Then your data set is ready to be log transformed. Your intensities are now log transformed. This is quick. This is super quick tutorial. So these are your intensity columns, 42 of them. And from here, you use regex to remove the name intensity. That's how Shivani did it. And you're able to clean that. You have beautiful names for your columns. After which you use your magic wand of expand site table. So remember, these is 42 columns and these are 41,000 rows. Expand site table should reduce the number of main columns and should increase the number of rows for the reasons I explained. So we use the expand site table and then you have main columns reduced to 14, matching the number of experimental conditions you had, and your rows has definitely increased. Mind you, a lot of them will be full of NANs because not every site is quantified in all possible forms, right? So a number of them, depending on the measurements, especially the higher versions, more than doubly phosphorylated versions are mostly NANs. So good place to filter in case you want to do that right away. After this is the right time to start annotating your groups. This is when you would use your categorical annotation and generate the classifications. Trying to think what was my idea last time. And that's when comes the favorite question: to impute, not to impute, what makes my matrix, what is the minimum number of valid values. Back to the rule, it has to be at least 50% or more, depends on your taste a little bit here. Quantify it in your random data set where you have no categorization, for example, or if you know a priori that there are different experimental conditions, in that case, 75 to 100% quantification in at least one group. So it's a function of reproducibility in one way or the other. So you filter for that, and now I'm back to from 125, 126 rows. When I apply this filter, I'm back to 52,000 events. And this is not because of, it has happened for two reasons. Number one, because we used the expand site table feature, which meant that a lot of highly phosphorylated versions of these sites, which didn't exist, and therefore they have to be NaN, they're also removed as part of this step. So that's why you see this filtering step has so much impact. It's always good to keep, as you notice now, keeping an eye on these tables and the numbers they move will tell you how your data processing is impacting overall your matrix. Then comes, I think now we would all agree that this is a reasonable data depth and filtering at which we can trust any imputation is not going to bring noise into the data. So it's only going to impute for wherever it was not identified, whereas somewhere else it was identified with enough robustness. And that's a good place to replace missing values. You can just explain it in this in the tutorial before, how you can do it with normal distribution and representing the lower abundance or the lower edge or detection of limit of the mass spec as your means to fill in those gaps. I'm sure a lot of you already know it doesn't make sense to put zeros there. Zero is much below than the detection limit, lowest detection limit of the mass spec. So at least that should be a minimal filter if you're putting the constant value. However, constant value is super artificial when performing any statistics. So for those reasons, this is the best method in our hands to do imputation. After which, we are now ready, and it's always good to look at your data before and after imputation and see the impact of imputation. You would see this looks scary. That is because, remember, our data set is composed of mitosis as a condition, which is full of so very exclusive events that happen during that stage that they are absent in the others, and therefore all those events kind of make this hump of imputed values in your data set, which again you don't have to worry about because you know you have filtered for robust presence in at least one group or biological condition. Does that help you understand when to worry about imputation and when not to? And it will get actually much clearer as we also walk through some of the downstream data analysis steps. Any questions? Either we need coffee or we are super clear. All right, after this, this is no different than a protein expression or an interactome analysis data set. You apply your ANOVA analysis. In this case, we've gone very stringent just to reduce the number of phosphosides, but it's always reasonable to start with the default parameters. And this step here would add an additional column in your categorical columns, which would be if the protein is, if the site is significantly changing. And you can use this in the next step to filter, based on this categorical column, the sites that are significantly changing. Again, you don't have to always do this step, but it all depends whether you want to perform your enrichments on only the regulated subset or do you want to do it overall, the total data set. So for now, for the ease and making sure that this session doesn't crash, I've reduced it to 15,000 sites, being very stringent. So using this filter, we found that of those 50K phosphorylation events, about a third passes that filter that we've just set. At this stage, you have another decision to make. Remember, I've not done any Z scoring, and this is a good place where depending on what your interest is. Do you want to look at intensities of these phosphosites, or do you want to just look at their extent of change? So for now, I've not done any z-scoring to see how the landscape looks on raw intensities, how differential is it. But this is a good place when you can do z-score and start doing your downstream clustering and enrichment analysis. Back to all sorts of fancy features in terms of annotations. They're all clubbed in this part here under modifications. These are all the annotations that you can enter. So expand site table we've already used. Linear motifs. So we all know that there are linear motifs associated with different kinases, for example, what do they prefer? And that information is already sitting in a data set, all well curated by Phosphosite Plus. Linear motifs as well, I believe, if it is not coming from UniProt. But there are many other features like, is this a site which is already known? By that, all we are doing is comparing it against the huge Phosphosite Plus data set. If any mass spec study before has already identified it, or the low throughput antibody-based methodologies have already identified it, when you do this cross comparison of your data set versus what is already known in the literature, starts to tell you how overlapping are you or how different you are in terms of the depth and the quality or the quantitative view of overlap. The next one is kinase substrate relationships. Again, there is a huge body of literature that has provided connections in terms of what is the upstream protein responsible for that modification in case of phosphorylation, which kinase or set of kinases are known to phosphorylate that residue on the protein of interest. And those relationships are sitting in a database very well put together by Phosphosite Plus. That's what we use to impute, again, those relationships into this analysis. The next one is sequence features. I believe this one is coming from UniProt. UniProt has many other sequence features in terms of also secondary structures, and I don't remember how many more features, but this is something that I use less, and therefore I wouldn't be able to tell more than that. Regulatory sites. So for me, this is another very quick go-to place. When I'm working with, 15,000 significant events as a PhD or a postdoc or a PI, once you have this matrix, that's when usually everybody says, good job. What do we do? What do we do next? Pathway analysis, you usually can talk about a little bit on that. But in case of PTMs, given these databases, one quick way of figuring out what's going on is go and see, is there a subset of sites which are already known to be regulatory in nature in terms of the biology that they induce. That's again very well documented on Phosphosite Plus. And I'll show you in one of the downstream analysis, there are about 495 sites of these 15,000 which are known to have clear biology associated with that. So if you wanted to get into quickly, is that subset made up of cell cycle based on maybe a 2/3 of that data set, then I know I have really impacted that well-documented pathway, and there are tools that I can quickly use to go back and track down the downstream biology. All right, and then there's this option of shortening the motif length. It's a flexible feature. So those of you who use sequence logo, it's again a matter of personal choice. Do you want to go 6 amino acid either side, or you think you want to go 10 amino acid either side? So this feature allows you to trim the sequence motif to the length that you want, and then start building up of those sequence logos. I have to say this is the first summer school where I've been able to complete all this annotation features despite the short length. So you would easily add all these features which are either coming from, which would require you to define the sequence window, which is coming out of your phosphosite, phospho-STY table, or again, if you wanted to say whether a site is already known, you will have to define what kind of modification is it, what is the protein that you're looking at, and the sequence window. These three features are then used to go back and track down the phosphosite plus to report whether it's known site or not. And therefore, it's important to upload all those columns, as I was saying. Next is to talk about the kinase-substrate relationships, would again require to define the uniprot column and also the sequence window.
Kirti, can I interrupt you here? We have a question.
Just a quick question. So all these annotations would have to be downloaded and placed in the conf folder, or
yes,
where do they have to go?
Yes, it's actually very well explained in the tutorial, the sheet that you have, but that's a very good question. We cannot provide phosphosite plus proprietary database data set to, otherwise that would have been part of all the Perseus downloads. You can go and download it from the phosphosite plus, easy to download, free for academics, pretty sure, or you have to get a license. But once you have that, all these features are directly being read from there. Although for the sake of this exercise, we have provided that folder on your USB sticks, but not exactly where it should be. So you have to do the moving part so you remember that we can distribute it freely. Okay. From here, these are all features, as I mentioned, for PTMs. But that doesn't mean you don't load all the protein features onto that. So you can do annotations based on Go enrichments, pathways, now also for kinase-substrate relationships. Are there motifs that are specific to any cluster or not? So after adding all these features, we can do our favorite clustering analysis, and I've already done it for all of us. This is a non-z-scored matrix. So if you look at the color scheme, these are really the raw intensities from the instrument, and it's actually pretty clear already here that things which are low here, high here, can be easily used to define some of the clusters, purely based already on the intensities because the differences are so strong. But I think if you added z-scoring, that'll be taking into account differential extents of changes and independent of the intensities. So that's why, even though I've done it this way, you should actually generate both the views as you analyze your data sets. So with this, you can start doing enrichments, and this is the place where I was talking about accounting for multiple representations of one protein just because it has more PTMs sitting on it by default, versus the other, which may have lesser counts of that PTM. Actually, based on this in-depth proteome and phosphoproteome in parallel, we learned that some proteins just like to be phosphorylated. They had more than 500 phosphosites sitting on them. And when we were doing enrichments, it made no sense. That's when we learned that, okay, we have to somehow account for this in our enrichment analysis. And how can you do that? Is by choosing one of these options. So when you say none, that means it's assuming that all your proteins are represented one time in your matrix. However, you can actually then say, I'm looking for these enrichments based on protein-centric view. So it'll then count multiple events as one protein event and take into account this multiple counts. So specifically for PTMs, you may want to use this feature to get the real signals. I'm actually pretty happy that I had never gone to this phase, this part of the exercise until the end, until so far. Yep, at the end of it, once you have all the orientations, this particular case is what we discussed in the lecture part of this, this talk. All the phosphosites that you see here are very exclusive to nocodazole-treated mitotic cells. And if you did not impute, you will be actually discarding this entire data set and all the meanings that you would have derived from it. So while it's good to be conservative, being over-conservative, you may actually miss the point. So impute, but logically, is the conclusion here. With this, I think I'm done with explaining all the bits of data analysis that are exclusive to PTMs. If there are any questions, I'm more than happy to answer.